The next sensor type we are going to learn is a biosensors. How biosensors are defined? Biosensors are basically a chemical sensing devices in which biologically derived recognition analyte is coupled to the transducer to measure the biochemical parameter of the analyte. Means whatever the biological sample that will be taken, that is called analyte, and that is reacting with a biochemical material of the sensor, which produces the response from the sensor, and that can be measured by the transducer connected to the biochemical receptors. Typical parts of the biosensors are the biological element. This biological element is basically the component which recognize the analyte, often called as bioreceptors. These bioreceptors are normally the enzymes, or they can be antibodies, they can be cells, it can be nucleic acids, or it can be aptamers. There are various biochemical materials those can be used for detection of the various properties of analyte. The analyte can be a blood sample, it can be a urine sample, or it can be other samples taken from the body. The second part of the biosensor is a transducer component. Basically, it is a semiconductor material or nanomaterial component which will convert the response of biological element into electrical signal. This electrical signal can be connected to the electronic system that can signal condition it for the appropriate display purpose or recording purpose. The bioelements, also called as bioreceptors, uses biomolecules from organisms to interact with the analyte. The bioresponse of this interaction is then converted to electrical signal by the transducer. As per the particular applications, the biosensors are also known as immunosensors, electrodes, resonant mirrors, chemical canaries, biochips, glucometers, and biocomputers. The biosensors elements are as follow. The variety of substance may be used as bioelements in biosensors. These substances are nucleic acids, proteins, including enzymes, antibodies. Antibodies based biosensors are normally called as immunosensors. Plant protein or lectin, a complex material like tissue slices, microorganisms, and organelles can be used for detecting the various properties of analytes. A typical construction and the parts of biosensors are shown here. The biosensor consists of a transducer which basically detects the various signal generated by biorecognition layer. These biorecognition layer elements are shown here as biosensor elements. The chemical reaction takes place here. If you see, the various forms of sample molecules are shown by different shapes. The biorecognition element can only receive the particular type of element only, and it will receive that element, and there is a chemical reaction takes place, and output of that chemical reaction is then converted by the transducer to electrical signal. The electrical signal will be amplified. Maybe it can be controlled by microcontroller and displayed on the display, or it can be recorded on a printer. The biosensor block diagram is shown in the following figure. See, whatever the analyte that is called sample, maybe blood sample for example, it is given to a biosensor, that is basically a biocatalyst. It will convert that sample to product, and during this reaction there are some parameters of the product, those changes, and those changes will be detected by the transducer. and transducer produce the signal in the form of electrical parameter that can be further amplified if required and displayed. Some of the biosensor types are electrochemical biosensors, amperometry biosensors, potentiometric biosensors, physical biosensors, piezoelectric biosensors, optical biosensors, and wearable biosensors. Various applications of the biosensors are common healthcare checking areas the biosensors are used, metabolic measurement for various metabolic parameters, those can be measured using biosensors. For example, measurement of insulin quantity and insulin treatment areas, the biosensors can be used. The clinical psychotherapy and diagnostic of diseases where biosensor finds the applications. The drug improvement and offense detection areas also finds the application of biosensors. Ecological pollution control also finds the application of biosensors for detection of pollution. A typical example of a biosensor is blood glucose measurement sensor. The blood glucose biosensor is used to measure the glucose content in the blood of a diabetic patient. In this sensor, the enzyme glucose oxidase is used as a catalyst, and that generally the reaction with the blood took place, which makes the oxidation of D-glucose, means the content of the glucose in the blood. The reaction is shown below. Say D-glucose plus H2O plus O2, that gives rise to D-gluconic acid and H2O2. D-gluconic acid changes the pH value of the sample, and here the oxygen content will be separated here. Those can be detected by using this glucose biosensor. The principle of glucose measurement is just a chemical reaction of the glucose oxidase with the glucose. It will takes place with the reduction in O2. It produces the H2O2 in the reaction, and there will be change in the pH value due to the gluconic acid. The measurement can be done by measuring the oxygen level by the two electrode. One electrode measures the ambient O2 level and other measures the O2 utilized during the reaction, and the difference of the O2 level is proportional to the glucose content in the blood. A typical blood glucose measurement strip is shown here. Here there are two electrodes. There is a exposure vent here, and there is a glucose oxidase gel. The blood sample is placed in this glucose oxidase. The chemical reaction takes place. The oxygen atoms are liberated, and those can be detected by these 2-point electrode cathodes. And the difference in the oxygen detected is proportional to the blood glucose present in the blood of diabetic patient. This way the biosensor works. That's all, friend, about the sensors. This ends unit number 5. Here there are some short answer questions and the long answer question for practice. So we will stop here. We will start unit number 6 in the next session. Till then, bye bye.